I'm Shannon Derman, doing my PhD at the University of Zurich, specializing in plant sciences, and I also form part of the EpiSeedLink consortium.
My specific role within the consortium is to look at the regulatory mechanisms that are behind plant priming. So what does it mean to prime a plant? Well, you can think of it as exposing a plant to a mild stress event that better equips the plant with mechanisms to survive a more stressful event in the future. But I'm specifically interested in seed priming, which is a pre-sowing treatment that allows for when the seed is planted to better withstand stressful events during germination and seedling establishment. We're interested in the epigenetic mechanisms that might be behind what we're seeing in the primed phenotype. When we talk about epigenetics, we're not talking about the study of the actual sequence that makes up the DNA molecule. What we're interested in looking at is how genes are switched on or off based on mechanisms that specifically do not involve changing the DNA sequence itself. I'm interested in finding out which of these epigenetic mechanisms are involved in seed priming. And once I know the mechanism that's involved, I can then ask the question of specifically which genes are being switched on or switched off in response to seed priming. We hope that generating better knowledge and understanding the regulatory mechanisms behind seed priming, we can lay a more scientific foundation of understanding these seed treatments. And we hope that we can work towards providing farmers with more stress-resilient crops in the future. If you found this research interesting, follow EpiSeedLink to find out what the rest of us are doing in the consortium.